Welcome back. We've been talking about selective permeability in membranes and how that arises from the structure of the membrane. And we talked about how the hydrophobic core of the membrane allows only certain molecules of certain size and certain chemical characteristic to pass freely. That's unaided. So what we're going to introduce today is the second aspect of selective permeability, and we touched on this already when we looked at facilitated diffusion and active transport. Remember, facilitated diffusion allows diffusion to occur down a concentration gradient across a selectively permeable membrane through a protein such as a channel. And active transport moves a solute up a gradient across a membrane, also using a protein with an input of energy. So we're going to expand on this idea of solutes moving across membranes with assistance. And this is where specificity arises when we talk about selective permeability. So those molecules or ions which can't be moved or can't move freely across membranes because of the chemical characteristics and because of the chemical characteristics of the membrane have to be assisted. And so assistance across the membrane to certain molecules is via the use of channel proteins and transport proteins. And these differ in structure and the mechanism by which they move solutes across membranes. Now, channels and transporters are specific for particular solutes. So a sodium channel will only allow sodium to move. It's constructed in such a way that only sodium can pass. Potassium, chlorine, calcium, other ionic solutes, they cannot move through the sodium channel, So this is where the selection also comes into play when we talk about selective permeability. One level of selection comes from having a hydrophobic membrane core, allowing only small nonpolar or small polar molecules to move across the membrane. We also get selectivity by using channels and transporters to help certain solutes across the membrane. If there isn't a transporter or a channel for a given solute, it can't cross that membrane. So membranes of certain cells can have certain proteins in them which allow only certain solutes to move. Another cell lacking a particular channel or transporter wouldn't allow that solute to move. And so different membranes can have different permeabilities. So let's look at some channels and some transporters. We'll start off pretty simple with channels. Channels are basically gates in the membrane. They are proteins which are transmembrane. That means they span entirely the membrane. They have a part on the outside of the cell or on the outside the membrane and a part on the inside the membrane. Now I just said outside of the cell, but don't forget there are membranes inside the cell. So there will be channels in the ER, there will be channels in the mitochondrial membranes, and so these will allow solutes to move down a gradient if they open. And there are lots of different ways in which channels can open and close. The cell can control that process. Now, transporters are a little bit more complicated. Transporters do one of three things. Uniporters, they're going to move one solute in one direction. So look at the name uniport. Uni meaning one or single, port meaning to transport, to move. A symport transporter will move one solute in the same direction. Sim means same, like symmetrical, that kind of thing. If we have an antiport transporter, now we've got two different solutes and they're going to go in opposite directions. So these antiporters will grab one solute and move it in one direction and simultaneously, or very close to simultaneously, move another solute in the other direction. Okay, so look at the names, and the names will give you some clues to what these things are doing. Okay, let's look at some of these in more detail. Here's a uniporter. Uniport, single solute, single direction. So the purple ball here is the solute, and the solute has been bound by a site in the uniport protein, and when the solute binds into this little pocket. Now go back to thinking about proteins and their three-dimensional structures. We talked about primary, secondary, and tertiary structure. Now this protein here is going to have complicated tertiary structure, and so there's going to be a little pocket just inside the protein, and a little cavity in the protein, and a specific solute will bind in there, and so the three-dimensional shape of the solute will bind into the three-dimensional shape of the pocket in the transporter. And when that happens, there's a change in the shape of the transporter, and so the solute will bind, the transport protein changes shape and flips the solute across the membrane. Now remember, the solute's going to be either charged or large and polar, so it can't diffuse through the membrane. So this protein kind of like just shuttles it across the membrane. So that's a uniporter, one solute, one direction. Now, in that case, the solute's going to be moving down a concentration gradient if we're talking about diffusion. Some of these processes can be active transport, and so it can start to get pretty complicated. Here's a symporter, two solutes, whatever the solute represented by the red ball is, and a blue triangle. Same kind of thing, a protein, complicated three-dimensional shape. The red ball binds in one site, the blue triangle binds in another site, and when both are bound, both must be bound. The protein changes shape and flips the solutes across the membrane. So you must have both solutes present for this to work. Okay, lastly, let's take a look at antiporters. Antiporters, remember, two solutes, but going in different directions. So here is what we have is a blue ball, blue solute, and it binds into a site in the protein, and that causes transport to change shape. It gets flipped across the membrane, and then a second solute from inside the cell or on this side of the membrane binds into the transporter, and when that binds, there's another change in shape. So this protein is constantly doing this. So you've got to have the blue ball solute on this side of the membrane and the red oblong substrate on this side of the membrane, so that you can get this flipping backwards and forwards. If you're missing a solute, if there's none of this red solute, then the transport protein is going to be stuck in this conformation. Now, a quick note about art. Sometimes the art makes it look as though, as this process is happening, that the protein is moving through the membranes. Now, proteins can move through membranes. They can diffuse around. We talked about the fluid mosaic model. So some of the membrane proteins are mobile, but this is meant to represent changes in time. So this is start, then this happens, then this happens, then this happens. This isn't meant to represent that the protein moves from this point to this point, and so on. Okay, this represents the next moment in time, and that's just the easiest way for the artist to represent it. Okay, so that's the second way in which we get selective permeability. Membranes have specific transport proteins which allow specific solutes to be moved across biological membranes. Okay, that's it for now, and I'll see you next time.